The overall goal of the following experiment is to use genome-wide barcoded microbe collections to understand gene-drug and gene-environment interactions in a highly parallel manner. This is achieved by screening the microbe collection for 5 to 20 generations of growth in the presence of a drug, using robotics to maintain the cells in exponential growth. As a second step, cells are collected, which allows us to extract genomic DNA from 4,000 to 6,000 individuals simultaneously. Next, PCR products comprising the amplified barcodes are quantified using either microarray hybridization or barcode sequencing in order to quantify the barcodes and thereby determine the abundance of each strain in a complex pool. Ultimately, results are obtained that show which barcoded mutants are sensitive to treatments and are therefore being depleted from the pool during the course of the experiment.
The main advantage of the technique we're going to see today is that rather than dealing with strains individually as colonies on plates or an individual liquid culture, here we can pool tens or hundreds of thousands of different strains and analyze them as one single pool.
So once we realized the power of this assay, we needed to automate it. And fortunately, I was working at the Stanford Genome Technology Center and was surrounded with engineers that allowed me to accomplish this.
Demonstrating the protocol today will be Andrew Smith, a senior graduate student in my laboratory, who will walk through all of the protocols and associated procedures, as well as Tanja Durbic, who will show us how to do the sequencing aspects of the protocol.
This protocol utilizes a library